at our second day, and it's my great pleasure and honor to introduce Professor Chantal Valeriani from Universidad Complutense de Madrid. She's going to talk about mechanical properties of active materials from suspensions of active Brownian particles to bacterial films. Thank you.
Thank you very much, Paulo. Good morning, everyone. My name is Chantal Valeriani. I first of all would like to thank the organizers. I don't know. Can you hear me? Oh, no, sorry, it was just somewhere else. Sorry. Okay, try again. Yes. So, yeah, can you hear me now? Yeah, maybe better. So I would like to thank the organizers for giving me the opportunity to present our work at this very interesting workshop. And today I'm going to tell you what we learned on active materials and their properties. I start by introducing, the team members of my group, very talented PhD students and postdocs who worked with me in the last few years. And today I'll be presenting you the work done by Miguel, Jose, and Boan. And this is the outline of my talk. I'll start by reminding you what active matter and active particles are, focusing on active Brownian particles. And then a small parenthesis on what to do if we do not know the interactions in our active matter system, and tell you what we learned on structure and mechanical properties of a phase formed by active Brownian particles, naming motility-induced phase separation, and on bacterial biofilms. So we can consider, Sri Ramaswamy and Cristina Marchetti, the founding fathers and mothers of active matter systems. Active matter systems are made up of units that consume energy to perform motion. Sorry. Why is it interesting to study such systems? It is interesting from a technological point of view, because one might think of assemble a functional material, a smart material, capable of performing tasks. But it is also interesting from a fundamental point of view, given that a suspension of active particles is driven out of equilibrium by its active nature. One might wonder, yesterday we heard already, sorry, I move here, because maybe I see it better. We heard already from Edgar, we heard talking about glasses. And so one might wonder, you're talking about an out-of-equilibrium system, is this like a glass? And actually the answer is no. An out-of-equilibrium suspension is not like a glass. The fundamental difference is the following. To prepare a glass, as Edgar told us yesterday, what people do, they perform a temperature quench on the system. They take the system and they cool it very quickly. If the system is a colloidal system, what you do, you apply a pressure quench. Take the system, you compress it very fast. Now, so you are applying an external field, temperature, pressure, to all particles in your system. This is very different with respect to having a collection of particles where each particle is an out-of-equilibrium system in its own. So maybe we can use some of the tools we learned in glasses, and that's what we are going to do, but the physics we are learning is slightly different. No, this doesn't, oh yes. So examples of these suspensions of active particles can be living or synthetic. Living active particles are birds, fish, bacteria. Synthetic ones can be a collection of active self-propelling colloids that can be made in the lab. From a numerical and theoretical point of view, many models have been tailored to study these systems. One can tailor numerical models to study swimmers in a fluid, to study bacteria, to study birds, but actually today I'm only going to focus on the model that was developed to study the behavior of synthetic colloids in a lab, so active Brownian particles. As Lisbeth told us yesterday, active Brownian particles can be mimicked. These are their two-dimensional equations by means of two equations, a translation and rotational equation of motion in 2 dimensions. This is the simplest case. We have spheres that self-propel with a constant active force. This is the active force that you see here that is directly proportional to the propelling speed. And then, given that these spheres that are isotropic in space propel towards a given direction, actually they are also characterized by a rotational equation of motion, where dr is the rotational diffusion coefficient inversely proportional with respect to this tau r, that is the reorientation time. This is the time already Lisbeth told us about yesterday, so the time it takes for a particle to change its direction of motion. How can we quantify activity? There are many ways, the persistence length, we saw it yesterday, But today I'm going to introduce you a slightly different one that is called the Peclet number. That is an 8-dimensional number that takes into account the propulsion speed and the reorientation time. So now that we learned what active brown particles are, one might wonder, how do they interact with each other? Because if we look at this equation of motion, if we forget about this contribution, this is nothing but the equation of motion of a Brownian sphere. So one might wonder, which interaction shall we consider between particles? And we could actually use our background knowledge in soft matter. In soft matter, people have studied colloids. Colloids are micron-sized objects that float in a solvent, and depending on their interactions, they can actually assemble in very different structures. They can form FCC-like structures, or when oppositely charged, they can form alternating cesium chloride-like structures, or they can form dense and dilute suspensions. So learning from soft matter, we decided to study colloids, active brown particles interacting by means of isotropic interactions that could be either attractive or repulsive. And we have learned the following. When interactions are attractive and the shape of the colloids was elongated, such as these dumbbells, what we did see was that particles were forming rotating clusters. So you can see these are experiments with bacteria polymer mixtures. These are in 3 dimensions. These are rods in 2 dimensions in here. And these are we just fine-tune the potential to make using a micellar forming potential. So in 2 dimensions or 3 dimensions with this, we saw particles aggregating, forming clusters, and due to their elongated shape, these clusters were actually rotating. Now, if particles were spherical and not elongated, we did see the formation of clusters, and these clusters were forming and breaking all the time. So just think of what we know in equilibrium. In equilibrium, a colloidal system made of attractive spheres would eventually phase separate into a liquid droplet embedded in a gas phase. Here, the liquid droplet is broken all the time due to activity. And these clusters form whenever there is a competition between attraction and activity and propulsion. So activity here acts as an effective repulsion between particles because they would like to stick together, but since they propel, they actually move away from each other. What happens if particles now are spherical, but they are repulsive? What we learned by my case as Julien Taillet 15 years ago was that if we have hard spheres, we do know that hard spheres either form a fluid, or if we increase a lot the density, they form a crystal. But these particles are very peculiar because this is the density-activity phase diagram. When the density is low and activity is low, particles are just at hard sphere suspension. However, if we start increasing activity and density, particles undergoes what Mike and Julien named motility-induced phase separation. You can see a few snapshots here. If I wouldn't tell you anything, you would tell me that this is something like a spinodal decomposition. And this is indeed the case. They observed this phase separation that is just due to activity. Particles are purely repulsive here. They do not attract to each other. So when a suspension is dense, repulsion induces the formation of MIPS. So activity acts here as an effective attraction. So both pictures exist, basically living clusters or motility-induced phase separation, depending on whether particles are initially attractive or repulsive. However, this motility-induced phase separation is not unique. The system is not in equilibrium. This is not a true phase diagram. And we can understand this because if we now consider stiffer interparticle interactions, basically WCA-like are softer, and then we can consider almost hard spheres-like. As you do see, this MIPS region here indicated with black dots shifts, moves. So basically, when the interaction is stiffer, the potential, this MIPS region shifts to lower densities and Peclets. Not only that, we are varying this Peclet number that depends on the propulsion speed and the reorientation time. It's not a unique way to define the activity. And if we now change the Peclet number by changing the reorientation time or the propulsion speed, as you can see, for the same system, we do get different phase behavior. Once more, the system is not an equilibrium system, and this is not a calculation based on free energy. We cannot easily define a free energy here. And how does a MIPS in a computer look like? A MIPS in a computer looks like this. So these are the repulsive sphere. They form clusters. The clusters start merging. And particles become kinetically trapped. And eventually, I just play the movie a bit further, they form this dense region embedded in a dilute region. And one might wonder, so how different is this MIPS from an equilibrium phase separation? And actually, the answer is it's very different. So we studied, that's just a liquid-vapor equilibrium. It's a Lennard-Jones in equilibrium. Nothing is active here. What we do, we switch on activity. What activity does, breaks completely the liquid phase, and we end up with an homogeneous sort of fluid phase. So activity acts as an effective repulsion between particles. If we crank up activity even more, what happens now, the system undergoes MIPS. But as you can see, this phase has nothing to do with this one. This is a phase separation induced by attraction in equilibrium. This is a phase separation induced by activity when the system is not in equilibrium. So to understand the differences, we started studying the fluctuations of this dense interface by means of capillary wave theory. We studied the interface by means of intrinsic density profile and computed the active force acting on each particle. And this is what you see here, the continuous line is just the average density profile dividing here the dilute and the dense phase. The dashed line is actually the intrinsic density profile that takes into account the nature of basically the local environment of each particles. In green is a contribution to the intrinsic density profile coming from particles with less than 3 neighbors, in blue with 4 neighbors, in red with 5 and 7, and in purple with 6. So you can see that the highest contribution comes from particles with 5 or 7 neighbors. And these particles are actually point defects. The system is in 2 dimensions. When computing the active force on each particle, again, if you look at the red curve, is the active force computed for particles with 5 or 7 neighbors. What we did learn was that, they were those contributing the most to the active force. So there is a strong force acting at the boundary between the dilute and the dense phase. So what these particles are doing are compressing all particles, the dense region. So there is an active force due to these particles with 5 or 7 neighbors that are compressing all others inside. Having learned this, one might wonder, okay, can we compute the surface tension by means of capillary wave? People in the past suggested that the surface tension for this system was negative. And actually, this is not true. What we did, we computed the capillary wave surface tension by means of mean square amplitude of the intrinsic surface. We get a wave vector-dependent surface tension, estimating its limits for low Qs. These are our results. So that's a capillary wave surface tension as a function of the active force. What you see that it's always positive, it is small, and it's non-monotonic. In order to understand the reason why it was non-monotonic, we computed the exotic order parameter as a function of activity, and we found that when the activity was low, the grain boundaries were percolating. There was a very low exotic order parameter. Increasing activity, the order parameter increased. So for high activity, there was no grain boundary, and this corresponded to a higher exotic order. So the slope change in the surface tension is actually due to the slope change in the order parameter within the dense phase. What happens now to MIPS when we start adding complexity? Meaning, again, coming from a soft matter background, can we study a binary mixture? And that's what we did. We started adding passive particles to a suspension of active particles. Everything is repulsive. This is, again, density-activity phase diagram. If you start adding passive particles, there's a passive particle concentration. What you do see is that this MIPS region starts shrinking. So the passive particles hinder the MIPS region. However, if there are not too many, we did see the same physics. Basically, active particles at the boundary of the dense phase, blue particles in the core of the dense phase. So few passive particles do not alter the MIPS structure. However, if we start adding 70% of passive particles, we still detect MIPS. But the structure of the MIPS has nothing to do with the structure of the MIPS we are used to. So fluctuations of these regions are very different. What we are working on at the moment is actually studying, again, the interfacial properties of this system of active passive particles, and we did find something very curious. So MIPS forms, but different from the purely passive case, the dense region now moves. So we observed a traveling band, and also we also observed the fact that passive particles are leading to much larger fluctuations than in the purely active case. What happens now in experiments? So far, I was telling you about computer simulations of active Brownian particles, whether purely repulsive or whether purely attractive. But what about experiments? Many people studied experimental systems in 2 dimensions, and that's why all simulations, most of the simulations are in 2D, because most of the experiments nowadays on active colloids are in 2 dimensions. And people did observe the formation of transient clusters in New York. This is from Paul Chaikin's group and David Pine. That's Clemens Beckinger in Germany. This is, again, Beckinger, and this is Cécile Cotin-Bison in Lyon. So they did observe formation of clusters, and actually they observed experimentally also the formation of MIPS, again in 2 dimensions. This is the group of Dirk Hartzell, Olivier Dauchot, and this is Cécile Cotin-Bison. So experimentally, people did see cluster formation and MIPS. But then the question is, how do colloids self-propel in experiments? It's a mess. There is a zoo of possibilities for colloids to self-propel. I mean, the main ingredient is that you need to break the symmetry, meaning if you have a colloid, this is an example of a catalytic colloid that is coated with silica and platinum in hydrogen peroxide. Given that platinum reacts with hydrogen peroxide, so you have a chemical reaction only on one side, and the colloid moves in the opposite direction. Same happening for photocatalytic colloids, or for these colloids that are dipolar Janus colloids. They are half-coated with a metal in an electric field. This, due to charge-induced electrophoresis, induces a net motion of these colloids. So one might wonder, okay, how can we mimic and understand the physical behavior of these colloids numerically? Is the active Brownian particle a good enough model? And if so, which interactions can we use actually to trying to understand the physical behavior of an experimental suspension of this system? So what people do in soft matter in equilibrium is well established. Since the confocal microscopy was born, what people do, what they can actually retrieve from experiments, the images and as a function of time. So at the end of the day, you have a series of images representing your system. So you have trajectories of your system. You have snapshots for different times. Then once you have the particles coordinates, you can compute the radial distribution function. From the radial distribution function, extract the potential mean force. So far, so good, but this holds in equilibrium. What happens now if your system is not in equilibrium? You cannot, strictly speaking, follow exactly the same route. It's not possible because the potential mean force cannot really used out of equilibrium. So what shall we do? Out of frustration, I started thinking, wait, we do have colloids trajectories. So can we actually learn forces from the colloids trajectories? And of course, the answer to this question is yes. So what we did, we teamed up with Dirk Hartzell in Oxford. He had colloids that are dipolar Janus colloids, so they are charge-induced electrophoretic colloids. And actually, these are movies from Dirk's experiments where particles were undergoing, here you see MIPS, here they were forming clusters, and here they were just in a fluid phase. So what we did, we built a graph of neural networks. We used a machine learning tool that we developed, which goal was to learn forces, not potentials, forces from particles' trajectories. So this is just, let's say, a snapshot coming from experiments, simulations, doesn't matter. So the ActiveNet is our tool, is a graph of 2 neural networks. Here you see in pink and in orange. So what we did, we sit on a particle, we draw a cutoff, we define a series of neighbors, assuming that the particle's motion is Brownian. Oh, sorry,
Just a quick question. The experiment is in 2 dimensions, right?
2 dimensions. Everything is 2D.
And can the self-propulsion go to the 3rd
No,
dimension?
never.
Okay,
No,
it's
only 2D.
Okay.
So self-propulsion is forced to be 2D.
okay.
I mean, of course, one can do everything in 3D.
Yeah, yeah.
But since in experiments they cannot tilt, so these colloids in experiments only propel in 2D, and they never, I mean, due to the electric field,
Got
they
it.
never flip because of the nature of the colloid.
Thank you.
So
Thank
the simulations are tailored to mimic these experiments. And so what we did, we have these graph of 2 networks. Yes, assuming that all particles are the same, that's an assumption, and there are no many body interactions. It's an assumption that can be released, of course, can be loosened, but for now, we only assume that interactions are 1 body or 2 body. What we did was the following. We built the first neural network, is described by this edge function, that what it does, it takes as input the positions of pairs of particles within this cutoff, and the output of this neural network is a function of the distances between these pairs of particles. The second network, what it does, it takes as an input the particles' position and orientation, and the output of the previous network, and the output of the second neural network is actually a predicted velocity. Why a velocity? Because what we did was minimizing the loss function computed as a sum of norm 1 of the difference between the measured and the predicted velocity, and we found the velocity that multiplied by the friction is the force. So in this way, we could find the 1 and 2 body forces acting on each particle. The 1 body force is computed from the node function setting to 0, the edge function, and the interaction force is computed from the node function removing the contribution from the 1 body force. And one might wonder, is this working or not? Sorry, I had an alarm. And how do I know whether this tool works? I use computer simulations. In computer simulations, I know what I plug in because I do it myself. So we simulated active Brownian particles, choosing the interaction potential and choosing the propulsion force. And then what we did, we plugged a series of snapshots, a trajectory, to the graph neural network tool. The graph neural network doesn't know whether the data come from computer simulations or experiments. And what we did, we learned that the tool works. So this is active Brownian particles interacting with a shoulder potential, meaning a potential characterized by 2 length scales, a short and a longer one. And we did repulsive, both repulsive. This is the 2-body force, not the potential, the force. It's a function of particles' distance, and you can barely distinguish what the network learned from what we input to the system. And then we said, okay, let's complicate our lives. Let's study a system that undergoes MIPS. Here, particles were interacting by means of WCA potential, whose shape is this one. And actually, here as well, you can barely distinguish the data from the simulation to the data learned by the network. And then one might wonder, okay, you can easily learn 2-body interactions. What about 1-body interaction, the propulsion force? Indeed, here, if you compare the dash to the continuous line, you can barely distinguish the input we gave to the particles with respect to what the network learned. Here we try to see the active force as a function of particles' orientation, hoping, as it is, that the active force did not depend on the particles' orientation. And so we moved to experiments, because this was our goal. Can we learn the interactions between active Brownian colloids in experiments? And the answer is yes. So we learned the active force as a function of particles' orientation, and we learned that the active force was isotropic. And then we also learned physics out of it. So the fact that the active force is directly proportional to the square modulus of the field amplitude is actually a charge-induced electrophoretic effect. And then we did learn the 2-body forces as a function of particles' distance that was a repulsive and an exponentially decaying function. For us, the most interesting result was that actually the network failed. When particles were undergoing a MIPS, given that our a priori assumption was that interactions were only 2-body, and MIPS is highly probable a many-body effect, the network, as you can see this line, was not working. Fine, that's perfect, because this is telling us that in order to learn many-body effect, we need to learn 3, 4 higher order body forces, and this is actually what we are working on at the moment. So we believe that ActiveNet can be used as a powerful tool to infer interparticle interactions, especially in experiments where you have no clue what's going on. And so now that we have characterized the structural properties of MIPS, what about their mechanical features? To study mechanical features, we applied an oscillatory strain in linear regime. Strain is directly proportional to stress by means of the shear modulus. So that's what we did. We measured the storage and loss modulus in experiments, in numerical simulations when varying at a constant density the activity. So here the system is in an homogeneous phase. Here it's at the boundary of the MIPS phase. Here it's deep into the MIPS phase. And it's very interesting what we get. So we get very different results because actually we have very different systems. First of all, the system behaves as a viscoelastic material. For large, these are G prime and G double prime, so the elastic and loss modulus as a function of the shear Peclet that is nothing but the shear frequency at the end of the day. So when the shear frequency is very high, all systems behave like elastic. So G prime is much higher than everything, G double prime goes to 0, so the system behaves more like a solid. Whereas for small shear Peclet, the system behaves like a fluid. Not only that, here you can see here the squares are the G double prime that circles the G prime. G prime grows faster here than G double prime, and this is a behavior of a Maxwellian fluid that I'm going to tell you about soon. So we did see that the system behaves like a viscoelastic medium, small shear frequencies, the viscous component wins, then there is a crossover and the elastic component wins. This crossover corresponds to different shear Peclet. Sorry, I don't know why I have an alarm again. And this actually crossover as a function of activity changes. So increasing the activity, these are snapshots for these different active Peclets. As you can see, they correspond to very different systems. So increasing activity, the crossover varies, the crossover increases because dense region starts appearing in the system and they shift the crossover up until the system up until MIPS takes place. When MIPS takes place, there is this drop of this crossover point because the system now is more of a solid. And if we now compute the viscosity, like the shear viscosity from the data we had, we observed something interesting. We observed that the system was a Maxwellian fluid. As I told you before, we already had hints before. A Maxwellian fluid is a fluid whose viscosity is first of all Newtonian, meaning it's constant, and then it decreases with the shear frequencies. So the system is a Newtonian and shear thinning system. Now, this for all activities, and this reminded us of what was a result that was found by Ludovic Berthier, Jean-Louis Barras, Matthias Fuchs, and Mike Cates long ago for colloidal glasses. Colloidal glasses, as you can see, they show all these features. So when the temperature is above the glass transition temperature, they are exactly Maxwellian fluids. So Newtonian and their shear thinning. However, when they are below the glass transition, they just show a shear thinning behavior. So again, there are similarities between active colloids and glasses, but it's not really a one-to-one map. But for sure, knowing, the physics of glasses can help us to understand the physics of these systems. Can we now make our life even more complex than that? So far, we were studying rheology of a material made of a dense and a dilute phase of active Brownian particles. 15 years ago, Dave Waits and Mike Brenner suggested that a bacterial biofilm is actually a soft matter material, can be studied as a cross-linked polymer gel together with colloids. So we were wondering, can we actually study the mechanical properties of such complex living system? But what is a bacterial biofilm? So we have to think that bacteria in real life are not happy swimming around because they're very small, they're micron-sized, and they can be attacked by pretty much anything. So their goal in life is to survive. So what they tend to do, as soon as they find a surface, they stick to it, they stop swimming, so they lose their flagella, and they start duplicating and at the same time producing this extracellular polymeric matrix. This the time span, as you see, is huge, and also the length scale span is huge. So bacteria micron size, a biofilm can be millimeters in size. And why do we care about understanding the mechanical properties of biofilms? Actually, to kill them. Because if bacteria are within a biofilm, they cannot be killed by antibiotics. So the biofilm protects bacteria against external cues. That's why if one finds a way to break these biofilms, then you can attack bacteria and kill them. So we started performing experiments together with my microbiologist friends in this setup that she calls a carousel. So we focus on Pseudomonas fluorescens. You have to imagine this cylinder is pretty big. It's 10 centimeters high, more or less. These glass cover slips are perpendicular with respect to the plane. And then what you see here in this drawing, in yellow you have the suspension of bacteria with their favorite food, that is TSB. In brown, in this sketch, is where the biofilm most likely take place, starts forming, that is at the boundary between the liquid and the surrounding air. So what we did, we started growing the biofilm in two different conditions. The first one, we just waited for the biofilm to form and started counting the number of cells formed as a function of the incubation time. This is hours, 24, 48, and so on hours. But then we started perturbing the system, meaning shaking the carousel you saw before, okay? The biofilm was forming again, but we observed very different features. So just by shaking the sample, the number of cells, so the results obtained from the shaking are in blue from now onwards, and in the static samples are in pink from now onward, okay? So shaking induces the formation of more cells. Not only that, if we now look at this, is just a side view of this glass cover slip I showed you before, and that's the surface coverage. We colored this with blue comassi, whatever, with a stain. What we did see was that when shaking, the biofilm was actually covering the entire sample, not just the liquid-air interface. So the surface coverage, again, was very different. And then what we did, we applied optical density, we measured with optical density the biomass, so cells and this polymeric matrix. And again, when shaking, we observed a very different behavior. So shaking, at the end of the day, was boosting the cellular growth and the matrix production. We studied by means of confocal light scattering microscopy the microscopic properties of this biofilm. What you see here is that the results under static, in red is a staining for the matrix, in green for the cells, and this is a combination of the two. That's a side view. And what you do see is that when the biofilm is grown under static conditions, the biofilm is thin, that's its height, heterogeneous and contain less matrix as compared to when shaking the sample. When shaking, the biofilm is thicker, homogeneous, and contain more matrix. To understand the structure, these were the structural properties. What about the mechanical properties? Again, we performed shear measurement. We used this rheometer. We inserted the sample in here. This is the plate. And we started applying an oscillatory strain and measured the shear in linear regime that gave us the shear modulus. These are experimental results for the stress strain in linear regime, so the dashed line. And we obtained the shear modulus. Oh, sorry, I went one. Sorry, I skipped. How can I go back? So basically, these are the results, experimental results, field symbols G prime, empty symbols G double prime. Just to remind you, G prime is the elastic modulus, G double prime is the loss modulus. Always, the field symbols are higher than the empty symbols, both in the shaking, here in black, and in static in red, meaning the material is always a positive material at all frequency. At some point, we see a crossover between these two, but for very high frequencies, and due to for a limitation of the instrument, we couldn't go beyond this point. And if we now choose a frequency, and we measure at a given frequency, G prime and G double prime, we observe that the material are completely different. So if we just focus on the red, in the static case, G prime as a function of incubation time, G double prime as a function of incubation time, the reds in both cases are more or less the same. So when in the static conditions, the material behaves like a soft material. When shaking, G prime, you see it here, is always higher than G double prime. So when shaking, the material is pasty. So the two materials are different. Not only that, if you now compare the blue and the pink, here the blue is higher than the pink, here the blue is higher than the pink. So shaking leads to a stiffer and a more viscous biofilm. How can we understand this behavior? Just remember what we learned. A biofilm, even though it's produced by bacteria, it's a complex medium made of cross-linked polymer gels and colloids. So what we did, we tailored a dissipative particle dynamics model inspired by previous works to actually try to understand the mechanical features of a bacterial biofilm. Our model is a basic model whose main ingredients are bacteria, polymers, and solvent. And basically, we learned from experiments that the concentration of polymers was not the same under shaking and static conditions, as I showed you before. So what we did in simulations, we added more polymers here than there. So the shaking system contained more polymers than the static system. And then, once having prepared the initial configuration with these elements, this is a bacterium, these are polymers, we started cross-linking polymers between themselves and polymers with bacteria. And this is an example of an initial configuration. And then what we did again, we applied oscillatory shear in a computer. As before, we applied an oscillatory strain and measured the stress to get the storage and the loss modulus. So this is an example of what we are doing numerically. The system is 3 dimensions, okay? Here you just see a side view because otherwise it was too confusing. And these are the results, G prime and G double prime as a function of the frequency. As in experiments, we observe that G prime is always higher than G double prime. So the biofilm is a pasty material. Now that this for us was, gave us confidence that the model was working. Computing G prime and G double prime as a function of frequency, we obtained the same results as experiments. So now that we could trust the model, what we did, we actually tried to use it to understand why this biofilm grown under shaking was very different with respect to the biofilm grown under static. So what we did with these computer simulations, we performed many simulations, varying the number of cross links between polymers and bacteria when the system was mimicked as if it was under shaking or under static conditions. And we compute the ratio between the stress, shaking, and static. As you can see, this function is a linear function. So the stress is a growing function of the number of cross-links, and shaking, at the end of the day, was leading to a larger number of cross-links in the polymer matrix. So this conclusion, for us, was not satisfying because we learned that the model was actually telling us that shaking was inducing a larger number of cross-links. But did we have experimental proofs? And to do so, to get experimental evidence of our hypothesis, we performed solid-state NMR under shaking and under static. Under shaking, as you can see here, there are more carbohydrates. There is a higher carbohydrate content. Translated into poor physical mind words means stronger adhesion. So our hypothesis was indeed correct. So shaking induced the formation of a stiffer and more viscous biofilm whose matrix is characterized by a higher content of carbohydrates. That means a larger number of cross-links. What we are working on at the moment is actually trying to see how much this effect depends on the experimental protocol, because what we are doing experimentally was a bit violent. We are removing physically the biofilm from this glass cover slip and inserting it into the rheometer. One might wonder how much these measurements depend on this protocol, and indeed they do. What we are doing now, they are repeating these experiments, but this time we are measuring rheology in situ. So with atomic force microscopy, it is indeed possible to measure G prime and G double prime without having to remove physically the biofilm from the glass cover slip. And this is already giving us preliminary results that look different when comparing our ex situ measurements to the in situ ones. And with this, I hope I've convinced you that suspensions of active particles and complex media produced active particles show interesting features. And I want to thank you for your attention.
Thank you very much, Chantal. That was wonderful. So we have time for questions.
In the case of your active colloidal particles, since you have a suspension and you're out of equilibrium, I would think that the hydrodynamic interactions would also be important, and those are very long range, so they would couple all the colloidal particles together. So it wouldn't be just two-body interactions.
Actually, they are not always, it depends on the density of the system. They might be important or they might not be. Also, the system is quite tricky because it's not 3D, it's 2D. So hydrodynamics in 2D is not the same as in 3D.
But I think it's actually a longer range.
Exactly, it's actually longer range.
So
yes.
Then it would couple more.
Yes, they could. We are considering also the effect of higher order contributions to the forces. But to a first approximation, it doesn't seem that it always plays a role. That's my point. My point is that, we said it, that we use the active Brownian particles model as a model to mimic real-life colloids, active colloids. To a first approximation doesn't seem to do a bad job. But one, of course, needs to add higher order terms. This can be done. The good thing about this graph of neural networks is that it's modular. You can add as many body forces as you want. But yes, I agree with you that there might be cases where hydrodynamics play a role. Yes.
Sorry, go on.
Good morning. Thank you for your talk.
Thank you.
In the beginning, you showed some data with electrophoresis experiments. So I think I missed it. That, I don't know, the particles or the colloidal suspension, are they charged in a certain way? Or they are free of charge, positive or negative charge?
Now, basically, these particles, they are coated. So imagine half of it is silica, is plastic. The other half is a metal. Okay? And then you have an electric field perpendicular to the system. It has been shown that at given amplitude of the electric field, if you set the frequency, these particles actually, due to this anisotropy, this is a charge-induced electrophoretic, is an induced charge, only on half of the colloid. And that's the reason why it's self-propels. Because the colloid is half plastic, half metal.
Yeah, because I was wondering if they are charged in a certain way, if this could play a role in the aggregation pattern, you know?
But actually, it's very funny because depending on the parameters of the electric field, these particles can either be a real dipole, so form a chain, but actually, in this regime, the experiments worked, the interactions were isotropic, and you can so they were not dipolar, and you can see it also by eye. These are movies from the experiments. These are not dipolar charges. They don't form chains. Real dipoles form chains. So they chose a parameter range where the interactions were actually isotropic, and you can see it also from here and here, because these structures so compact cannot be made by dipolar interactions.
Thank you.
Thank you.
Thank you. I was wondering, so you used your AI generated learning process for learning properties of this let's call it the simplest of all active matter models, because you get rid of basically all the chemistry, right? There is only like the electric double layer forming and deforming.
Yeah.
Do you think there's anything that you could learn from more complex systems?
Yeah.
Any parameters?
Yeah. We are actually using this. This was just a work that actually took us ages to publish. That's an example, as Edgar was saying yesterday, never give up, because people, now they love it, but it took a long time for people to understand, to appreciate it, let's say. But of course, now you can go wild. So I have experimental data from research groups that have particles that are actually catalytic. So, sorry, I'm just going to show you basically catalytic particles. Do I have them somewhere? Sorry, just looking. Where did I see them? Okay, whatever. Here. So we have experimental data from Lucio Isa at ETH group. These particles are photocatalytics. So they are titania coated. You shine UV light in the presence of hydrogen peroxide as propel, and we are using this method to learn the interactions in this case. And actually, we are also playing with particles that are anisotropic, not even particles. We are applying this method to ants. Ants, you know the ants? The insect. Because there are interactions there, and you can, of course, there is a lot of work we are doing because it's not so trivial to extend the method to anisotropic interactions, anisotropic shapes, but can be done because the the method is modular, as I was saying before. So you can just add more modules and you can learn different, as usual, I think that machine learning is a great tool, but you always need a physical intuition. It's not a black box that you press and it gives you whatever. No, it doesn't work like this. You need, you need the physical insights to understand what you're doing, but of course the method can be used to many more, to different active colloids.
And can you give me some examples of parameters that you can actually learn from the interactions?
I told you, we are playing with these particles. Lucio Isa's particles are photocatalytic. We also have particles from Jeremy Pallacci in Austria, and his particles are catalytic. So, but this is an ongoing work. You can use that, but you don't add anything. Let's say, it's not that you change what the network needs. The input of the machine learning is just particles, positions and orientations, nothing more, okay? And you get positions and orientations of all these colloidal experiments, okay? You don't need anything else. We can talk more about it.
Thank you for your talk. I was wondering if you could be able to measure the electrophoretic coefficient or self-electrophoretic coefficient using your method.
The thing is that then you are assuming already, my goal was to learn interactions without knowing forces. Mm-hmm. So if I know the forces already, but if I learn electrophoretic forces, yes. Okay. The thing is that there is another step I didn't mention, but because this was just, my goal was just to present you the method, but the goal was, the talk was not on the method. Another thing I didn't mention you was, when you get something like this, okay, one might wonder, okay, what is this? There is a way to actually backtrack. Once you have a functional form to learn, it's called symbolic regression. Once you have a functional form, you learn by means of symbolic regression. Once you have the output of your machine learning, you learn the functional form by means of symbolic regression. So there is a mathematical robust formalism to learn properly, to understand which is your force, and then from this you can go wild. But at this stage, what I showed you here, you wouldn't know what kind of force is this. Here we know it because we knew, but even in the exponential, in the experimental case, what I showed you here, this was just our experience. So we knew that this was an exponentially decaying function. But formally speaking, one should apply symbolic regression, that's what we are doing now, to get the functional form. And then once you get the functional form, you can compare it to existing functional forms and get parameters out. That's what we did actually here, because we got the parameters from this exponential decay.
Okay, thank you.
Thank you for the very nice talk. I'm just wondering, how exactly do you compute these phase diagrams for the non-equilibrium systems without a free energy profile or a potential mean force? Do you have at least an order parameter or something like that?
Yeah, sorry I didn't mention it. So when you get to any of these phase diagrams, I would call it state diagram because, what you do in practice is just one way is to compute the local density, okay? You compute the local density in your system, and then you compute the local density distribution. If the system is in an homogeneous phase, this local density distribution is one peak, okay? If the system is phase separated, the local density distribution is two peaks. So that's what people do, okay? But of course, it's not a thermodynamically robust calculation because you cannot apply thermodynamics here.
Yes, but it acts like a order parameter.
Yes, local density,
okay.
Yes.
Thank you.
Thank you.
I have a last one, if I may. In the experiment where you showed bacteria in clusters rotating, right? So the simulations that you compared with are dumbbell simulations.
Yes.
Why don't you get like flocking behaviors or something like that as you have in other experiments with elongated bacteria?
I mean, the system, because there is, sorry, I didn't mention it. Oh, I think is it written? But there are attractions between elongated objects, bacteria or dumbbells, doesn't matter. So when they come together, they stick.
Ah, okay.
Okay. So there is an explicit attraction.
So it's not like Peruani, like self-propelled rods.
No, these are colored polymer bacteria polymer mixtures that in experiments they stick because there are attractions, and in simulations we played with some sort of a Sakura-Osawa model to mimic the concentration of polymers. Knowing the radius of gyration, we knew the attraction depth and strength, and so we performed simulations with a short-range attraction, as in experiments where there is an effective attraction due to the presence of polymers.
Yeah, sure. And just very quickly, can we speculate about why the bacteria in the shaking experiment produce more
because
polymers?
they want to stick more.
They just want to stay around.
No, because they somehow, it's amazing, but they change their phenotype in order not to be washed away. Yeah. So they start producing a material that is stronger, stiffer, everything, just because otherwise, the shaking will just wash away the biofilm. Yeah. So that's why they change their machinery to produce different biofilms.
Yeah, that's nice.
Thank you.
Okay, so let's thank Chantal again.
Thank you.
Thank you.